Today we are going to revise ETC. So ETC is actually a series of reactions or a series series of oxidation reactions in which substances like glucose are oxidized into carbon dioxide and water. But this is not a single step, it is actually a multi step reaction in which the intermediates donate electrons. There are various intermediates, so and so forth. They donate the electrons to uh, certain coenzymes, or for example, NAD, NAD plus, FAD, etc., etc., and they further transport these electrons ultimately to oxygen, which combines with protons to form water. Now, during this process of passage of electrons from the intermediates of oxidation of glucose to carbon dioxide and water, the electrons are transferred through various steps ultimately to oxygen, and in this, the electrons, as they are passed, they actually lose their energy. They are losing energy, and in this process, the energy which is released is actually being used to transport protons or H plus, across the inner mitochondrial membrane. Inner mitochondrial membrane. And this movement of protons will then be used for conversion of ADP and inorganic phosphate into ATP. So ultimately, the aim is to produce ATP. So the glucose we ate got oxidized into CO2 and water, and through some of the intermediates transferred the electrons to certain coenzymes, and which further transferred it to oxygen, making ultimately making water. And during this transfer of electrons, there was loss of energy, which energy was used to pump hydrogen ions or protons into the inner through the inner mitochondrial membrane, and ultimately these protons were used. The proton gradient produced was used as the source of energy to produce ATP. Now, if we begin with the ETC, so firstly, talking about mitochondria, the place where this is actually happening. So it looks somewhat like this, and the inner membrane has folds called cristae. So this is actually a double membrane structure. This is the outer membrane and this is the inner membrane. Alright, it has folds. These folds are known as cristae, and the outer membrane is has a property that it is freely permeable. Freely permeable to most ions and small molecules. Not large molecules, of course, but most of the ions and most of the small molecules can easily pass through this outer membrane. So it is freely permeable, while the inner membrane is not. The inner membrane is actually impermeable to most of the ions, including even the protons and other molecules like ATP or ADP or pyruvate or any such molecules. So it is really very, very impermeable. Hardly any substance passes through, but the outer membrane is freely permeable. So what happens is, if we have to move anything across this inner membrane, we would need some special ports or channels. So these inner membrane are going to have some special channels for the process of transport of the substances. Now, the inner mitochondrial membrane is rich in proteins. Rich in proteins. All right. So coming to the matrix. Matrix is the part in the inside of this mitochondria, which would be this space. So here the substance which is present, the gel-like substance which is present, is known as matrix. This is also rich in proteins. Just a second. This matrix is rich in proteins. Now, what do you think these terms rich in proteins means? They are actually being referred to this protein, and this protein are actually being referred to enzymes of the electron transport chain. And not only electron transport chain, various other processes that occur in mitochondria, but they are basically enzymes. What else the matrix would be containing? It would contain the see, if electron transport chain is occurring in the matrix, the substrates required in this process would surely be present here. So what is required in this process? It is these coenzymes or electron carriers like NAD plus and FAD. So these would definitely be present in the matrix in order to electron transport chain to occur. Also, the ultimately the thing being produced is the ATP, for which we require ADP and inorganic phosphate. So we would also have ADP and inorganic phosphate. In addition, it would also have its own DNA and RNA, because it is a special type of cell organelle which also has its own DNA plus RNA is always there. Now, if we specifically move our attention to this inner membrane, let's make it the main heading. Inner mitochondrial membrane. So it actually contains 4 different complexes, which are very simply numbered: complex 1, 2, 3, and 4. So what these do is they are involved in the transfer of electrons from the intermediates to oxygen. So the electrons go from these complexes to further and ultimately to oxygen, and this change happens through some mobile electron carriers. What are these? We'll see. Drawing this, we have this complex 1. It is known as NADH dehydrogenase complex. What happens here is that the NADH plus H plus is acted upon by NADH dehydrogenase enzyme, which gets converted to NAD plus, and these two hydrogen are transferred to a compound known as FMN. Now, what FMN would be converted to? FMNH2, and it will further give its hydrogen ions and electrons to the iron sulfur complex. Alright, so if we want to have a look at these structures, so this thing is the NAD plus. It contains nicotinamide, adenine diphosphate. Nicotinamide adenine dinucleotide. Nicotinamide and a dinucleotide which has this adenine group. So this accepts hydrogen and 2 electrons to give NADH plus H plus. This second H plus remains combined with this. And the FMN is the flavin mononucleotide, which looks like this. It is the flavin group, and this is the mononucleotide group containing sugar and phosphate. So flavin mononucleotide gets converted to FMNH2. Ultimately, the electrons are transferred to the iron sulfur center, and from iron sulfur center they are then transferred to a mobile electron carrier, which is coenzyme Q, which is also known as ubiquinone. So this is a quinone, as its name can suggest. It's a quinone derivative. So quinone was actually something like this, so this compound uh, got some extra group. Yeah, here we have quinone. So this thing was quinone plus some extra groups like this isoprenoid tail, and it formed ubiquinone or coenzyme Q. Note that the word ubiquinone is here because this enzyme is said to be ubiquitous, which actually means very abundant. So because this enzyme is very abundant, so from ubiquitous it came to be known as ubiquinone. And similarly, this would get reduced to COQH2, or which is also known as ubiquinol. If you have some idea, you will know that ketone gets reduced to alcohol, and that is what is happening here. A ketone is getting reduced to alcohol. While in the same step, we have this complex 2, which is known as succinate dehydrogenase. Now it has further FAD and the same iron sulfur center. What happens here is the succinate dehydrogenase, as the name says, will oxidize succinate. And when succinate is oxidized, it forms fumarate. See, succinate is this compound. Now, when this is oxidized or a hydrogen is removed, it forms a double bond here, and this structure is known as fumarate. So in this conversion, the two hydrogen ions released are transferred, two hydrogen ions and the electrons released are transferred to FADH to form FADH2. Then the electrons are further transferred to iron sulfur center, and ultimately they are transferred to the same ubiquinone. So from complex 1 as well as complex 2, the electrons are transferred to ubiquinone, which gets reduced to form ubiquinol. But one difference to notice, that in complex 1, while the electrons are transferring from higher energy to lower energy, there is a transfer of 4 hydrogen atoms, 4 hydrogen ions or protons from the matrix into the intermembrane space. From the matrix into the intermembrane space. So the hydrogen ions actually get collected in this area. The 4 hydrogen atoms are transferred from here to here. So we had these ports here through which this hydrogen was transferred, as inner membrane is actually impermeable. Now, from where did we get the energy to transfer these hydrogen ions from the ports? It was actually when these electrons transferred from their high energy state to low energy state. Now, when ubiquinone or now ubiquinol has the electrons, it will transfer the electrons to complex 3. Now complex 3 will have cytochrome B, again the iron sulfur center, and cytochrome C1. So what actually is cytochrome? Um, Cytochrome has just a structure like heme group. If you remember heme, it has a porphyrin ring with iron. So the structure you can see right now is porphyrin ring, and when this iron is attached, it becomes heme group. So this cytochrome has this porphyrin ring and iron. But the difference here from hemoglobin is that uh, the iron here present in this can reversibly change its oxidation state from +2 to +3, and during this change uh, is actually how the electrons are transferred. So the electrons are actually transferred from ubiquinol to cytochrome B. So the oxidation state would go from Fe plus 3 to Fe plus 2, which are then transferred to the iron silver center, which are then transferred to cytochrome C1, which then transfer these electrons to a another substance known as cytochrome C. Note. This is a mobile electron carrier. It is present in the intermembrane space. And it is carries the electrons from cytochrome C1 to complex 4. Now this complex 4 has a copper center, cytochrome A, another copper center, and cytochrome A3. This complex, the previous complex was known as cytochrome BC1 because it had cytochrome B and C1, or cytochrome C reductase. And the complex 4 is known as cytochrome A3 or cytochrome C oxidase. Now what is special about this complex 4 is that it is the only electron carrier in which the heme group, which is present in the cytochromes, it has an space for oxygen to bind. So the oxygen will bind here, and the electrons which were transferred from cytochrome C to the cytochrome AA3, and with the help of these copper centers, the electrons and the free hydrogen ions or protons are brought together, and there is formation of water. The oxygen is reduced to form water. Electrons are moving from copper center A to cytochrome A to cytochrome A3, which is associated with the copper center B. And this ultimately transfers the electrons to oxygen, which binds with protons and gets reduced to water. So this covers this part of the process known as electron transport chain. Now, how this lost energy is used for hydrogen transport, and conversion of ADP to ATP is a different topic. Now, something special about this ETC is that there are certain inhibitors which, like, they act anywhere between the process, maybe like here or here or maybe here. So anywhere they act, and what they do is they stop the flow of electrons. So if electrons are not flowing, they would not reach the oxygen, and which will not cause the transfer of protons, and which will ultimately lead to no ATP production. So it's actually the ATP production will stop due to these inhibitors. So what are these inhibitors? For example, the amytal and rotenone. These two act and stop the electron transfer to ubiquinone. That is, they act in this process, they interrupt this process. So what they are doing is complex 1 to coenzyme Q, they stop this transfer. Secondly, we have a compound known as antimycin A. What this does is it will act between complex 3 and cytochrome C. Complex BC1 and cytochrome C. So this is actually complex. It is stopping the electron flow from complex 3 to cytochrome C. Also, we have another set of inhibitors known as cyanide, which is a very popular poison. Also, we have carbon monoxide, hydrogen sulfide, and sodium nitride. And what these four things do is they act here. That is, they stop the electron flow from cytochrome 4 to oxygen. Complex 4 to oxygen. So the complex 4 electrons aren't transferred to oxygen. So these are the three types of inhibitors which can interfere with the electron transport chain. We also have something known as reactive oxygen species, that is ROS. So what happens is, if anywhere during the ETC there is leakage of electrons, so the oxygen is not fully reduced to water and it forms compounds like superoxide, hydrogen peroxide, or hydroxyl radicals. Now these substances can damage the DNA. They can damage the DNA, they can damage the proteins, and they can cause lipid peroxidation. So this is not something we would want, right? So what is done here, there are some enzymes like superoxide dismutase, the short form is SOD, and another enzyme like glutathione peroxidase, and another enzyme known as catalase. So what these enzymes do is they act as cellular defense against these ROS. All right.